Well, so can you hear me properly?
Yes, yes.
Okay. So first of all, thank you, Sadhin, for introducing me. But you praise me very much, but I'm not at all like that. So, so dear friends, today I'm really grateful to the organizers because in this pandemic situation, they are trying to provide a platform which is giving some food for our mind, and they are giving me the opportunity to present my research work in front of you. So today I will discuss on super resolution microscopy. You can assume it as a love triangle of biology, physics, and chemistry. So there are slides which may be a bit difficult, but I will try my best to explain because most of the participants are here school students, right? I like them because they are the young mind. And initially, it's remember my school days. Okay, so are you familiar with this cartoon? So this cartoon, I have collected this cartoon from Abul Tabul. It's my favorite cartoon from Abul Tabul. So if you uh, want to develop any technique, you need to have a definite question, right? So in 16th century, when people are trying to search new things against the orthodox idea of the church during the Renaissance, then they have a several questions. So there was no separate biology, separate chemistry, mathematics, physics. Nothing was separate. So some people, they are trying to observe a few specimens which is very small, and they were trying to investigate that in a magnified view, but they don't have the tool at the time. So then physicists came, well, yeah, we are there, we have the beautiful lenses. So in 16th century and at the early 17th century, Galileo Galilei gifted us some beautiful lenses which were eventually converted into the telescope. But Liberty, he was playing with those lenses to develop the reverse thing. That means whether we can observe a small specimen in a magnified view. And he was the person who has pioneered, who was the pioneer of the modern microscope. And let me tell you, this is the first commercially available microscope. I think all of you are familiar with this picture and with this instrument, because in most of the school can buy that, and it's cheap, but it's easily available. So this is the first uh, commercially available microscope made by the Nikon. Well, so let me speed up a bit. So what's the basic principle of the microscope? So you want to observe a specimen in a magnified view, right? So you need a light source. So you have a light source. Most of the light source are in diverging condition. So you need to focus them to the sample. So you have to use a lens that's called the condenser lens, which will condense the light. Clear? And then your sample will absorb some light, and rest of the light will be transmitted, and the transmitted light will be collected by another lens, that's the objective lens, and you will get an transmitted image in your eye with a magnified version. That's called that was the primitive or the first microscopic mechanism which people can use easily. Okay, let's see the basic principle. So that was, I'm trying to, well, so that's the structure of a modern microscope, where I placed the sample and I need to provide the light. Well, let's see. Yeah, the light is coming, it's the sample are absorbing light, and some light will be transmitted and you can observe in a magnified view. So that's called the transmitted light microscopy. Suddenly, after a few years, people observe that some samples are there or some specimens are there which can provide it when you excite the sample with light, it can give you back a light with different color. That's called the fluorescence. So can we have a look? Yes. Okay. So I'm trying to excite this sample in different way. So we have a different light source. We are trying to excite it. Yes. And the sample is giving back a light with different color. Well, so that's called the fluorescence microscopy. So what's the difference between them? So so this is a image of villa cell under the microscope it's a transmitted light microscope. So you can visualize the image in a negative manner. That means those samples which are not, which are getting absorbed by the, which are getting absorbed and in the slides, and some of the component of the cells, they will absorb the light and emit the rest of the light. So you will get a negative impression of the light. But here, the same cell will give you a different color of light. Well, so that's the difference. But the beautiful thing is that as cells are giving different type of colors, so you can easily observe them with different colors, right? Because all of the component will not have the same intensity of the light or intensity of the fluorescence. So you can compartmentalize the whole specimen, right? So that's the basic beauty. So that was the image from transmitted light microscopy. That was the image from fluorescence microscopy. Well, that's fine. But can we combine them? Yes, we can combine them. How? Just you set up different sources, light sources in different position. So I'm giving you a scheme of a modern microscope, which we normally use. So it has two light source. So from the top, you can blaze the sample with white light. That means for the to get the transmitted image, and you can blaze the samples with the laser light to get the fluorescence image. Okay, so that was the track for the fluorescence, and that was the track for the bright field. Even you can call it a bright field image. Okay, so what's the basic principle behind that? So you have to use a dichroic mirror for that. What is the function of dichroic mirror? It's a selective mirror which can reflect the light of selective wavelength and which can pass the light of different wavelength. Let's say you have a excitation light which can reflect through this dichroic mirror, and the sample is emitting some different color of light which has less energy or the lower energy than the excitation one, that will be emitted and that will be passed by the passed through the dichroic mirror, and you can collect it and even you can observe it. Okay, so that's the basic principle. Don't go for the engineering. It seems to be too hard, but that's not the problem. So basic principle is that. Okay, I'm trying to explain what is the basic principles of fluorescence. So any of the molecule, if you excite with any form of energy, here we are interested in the photon, that means the light. If you excite the molecule with light or the photon, it will be excited into the higher electronic level. That's very usual. And the normal tendency of the molecule will be to dissipate their energy in terms of other energy. So they normally dissipate their energy in terms of heat, which is a non-radiative process, and normally some molecule they prefer to dissipate their energy in terms of radiation, and that's called the fluorescence. That's the basic explanation. maybe I'm trying to convince you in a simpler way, okay, in a very simple way. Well, so the thing is that now you have a, your sample is giving you back a light with different color. That's the fluorescence. So which type of sample they are? Well, so when people are trying to develop the fluorescent microscopy, so they need for a fluorescent molecule. And now physicists are unable to provide those type of different colors. But chemist was there, and they were providing different color of molecule because they can generate it. So now there is a collaboration between physics and chemistry, and the question was from the biology. So chemist had lots of molecule, and that's one fluorophore. So if you carefully observe, yeah. So am I audible?
Yes, yes, yes. You are
Yeah.
audible, correct.
Yes.
Okay.
Yeah.
So so let's say this molecule is Alexa fluor 488. Why 488? Because it can be excited with the 488 nanometer laser or light, and that will give you the fluorescence with different color. Well, so more the number of pi conjugation in the benzene ring, the whole conjugation in the whole conjugation, then you will have a less energy fluorescence. That means you will observe a fluorescence in the visible range. So that is 488 with this existing condition or existing pi conjugation. If you increase the conjugation, then your energy will be lower because electron will get more room to roaming about and for the delocalization, and it will give you fluorescence in the visible range. So what is the visible range? And let me tell you. So it's normally from 450 to 790, and we normally try to use those wavelength because, or within that wavelength range, because you can easily detect them, and normally the biosamples are um, so light is fatal to the sample, any type of heat energy, because it's generate heat. So with those lights you can prevent the damage of the sample. So normally, and you can easily detect them. So normally people use that. Well, but the problem is that if you use an external compound like that, that's a different chemical compound which you are going to devise. But why cell will accept that? Because cell has itself a biocompatible environment, then do not allow the foreign particle into their microenvironment easily. So people were trying to deliver some fluorescent molecule within the cells, but the cells are not taking that, or it's assume, or you can assume cells are denying that by so there is a term. Okay, so are you familiar? I think you are familiar with the autophagy. So autophagy is nothing, it's a programmable cell death. So cell can program their death, right? It's a suicide of cells. So if you apply some fluorescent molecule, sometimes cell suicide makes suicides. So that is very fatal to the sample when because you are trying to observe the cells in a living condition. So people has discovered people who are searching for a molecule which is already within the cells but fluorescent, and those three scientists in 2008 were awarded for the Nobel Prize for the discovery of the green fluorescence protein, which was purified from jellyfish, so Aquaria victoria. So some of the jellyfish probably you have already observed in video or in the museum or maybe in the zoo, that they can give you back a nice fluorescent light. So some of the jellyfish can give you different color of protein. One of them is GFP. But the problem is that those protein have some advantage, also some disadvantage. So what is the advantage? Advantage is that it's biocompatible, so you can easily deliver it to the cells. right? But the disadvantage is that it's extremely photosensitive. If you try to image the cell for a long time, it will be damaged because they are very much sensitive to the light and they will misfold. And if the misfolding occurs, then that protein will not be fluorescent. So what was that? What is why that is important? Okay, so now scientists has developed uh, some mice. It's called transgenic mice or rat, and you can easily tag those mice with your favorite color, with your favorite protein. Like let's say it's GFP, and I'm showing you it's some rats. So they are transgenic rat. They are tagged with RFP, red fluorescence protein. So let's see how they behave under the microscope. Yeah, they are working. It's nice, right? So, so those mice are those rats are very useful for the study of organ transplantation and even in the neuroscience studies. So now you can tag your rat, your any sample. You can tag your any sample with the cells with a GFP. Well, but the problem of the GFP or any fluorescent protein is that they have less photon count and they easily got breached. So that's why you cannot use that. So people again started to rely on the dyes. Now the question is how you will tag your dye selectively in your biosample, because it's a foreign compound and you have to tag it selectively, because if you have a free dye in the sample, then it will give you a false impression, right? Because your fluorophore is the representative of your biomolecule, right? It's the representative of at any particular biocomponent, right? So let's say I'm trying to tag a DNA. So chemist has fantastic techniques. So they have the name reaction. You are familiar with the name reaction in +2 level, right? So every dye which we normally select for tagging, they should have a group called malimite. So why that has been used? So malimite can undergoes a substitution reaction which can form a stable covalent bond between the dye and the biomolecule. So now using a substitution reaction between the malimite and the thiol, you can make a stable tagged biomolecule if you want. And you can also tag the protein because the protein itself has a cysteine residue, right? So cysteine residue has thiol, and you can easily tag the thiol with your particular dye using the malimite reaction. Even also people use the succinamide version. So succinamide, what succinamide do? Succinamide interact with the amine, and protein has lots of amine, and you can selectively place your amine by genetic cloning or molecular cloning, and you can tag your protein as per your requirement. Okay, but the most useful approach is that antibody tagging approach. So let's say you want to observe a specific organelle of a cell, let's say mitochondria. So how you will observe? So you have to select a small protein which can specifically interact with the mitochondria, and you can tag the protein with your particular color, I mean the dye, and you can tag the other component, let's say nucleus, with other dye. Okay, and then you can individually identify the mitochondria and the nucleus. So that's the basic facilities of antibody tagging. So it's very selective, and I'm giving you an example. So that is a 18F tagged antibody fragment, and that was injected. Okay, sorry. So that was injected into a mice engrafted with pancreatic cancer, just to observe where the pancreatic cancer started first. So you can, so we have labeled, so if people had labeled this mice with the 18F dye, and which is tagged with the primary antibody. So antibody tagging approach is another approach to tag biomolecule. But the thing is that most of the examples which I already provided is of in the order of millimeter or in the order of, let's say, centimeter. It's not that much microscopic, right? People were happy with that at that time, but problems started when scientists were trying to observe that how cells crosstalk with the others. I mean, within that. So any signal that will be transported within the cells, how cell transport that signal to a particular component or particular compartment. Let's say you have a signal, you have to, or you have a signal, mean, let's say you have a protein, you have to deliver that protein into the mitochondria. So how cells regulate that? That was a prime question at the time. And with the existing setup, people can image this type of cells. That's a HeLa cell where you can easily tag, as I already discussed, you can easily tag the nucleus with different color. It's a blue color. Normally people use DAPI for that, and you can tag the microfilaments or microtubules with other color. Okay, let's have a close look. But when people are trying to observe how the cargo is getting transported into the cells, they found that some cargoes, some cargoes are walking through the microtubules. They are using the microtubules as a track to transport their signal. You can easily observe it, right? So they were happy. Okay, it's now we know the role of the role of the microtubules, what they did. So what they do normally, they use the microtubules to transport their signals into particular component. But how they are working? That's a prime question, because I want to change the track. How I will do that? So always scientists have different questions. Okay. So they are not at all happy with the existing one. So anytime you can observe this, you want to observe that particular track or that particular working of the protein on the microtubules. But those are very tiny. You can observe it's very tiny molecule. So you need to have a resolution so that you can decipher the track properly. So that's how the concept came to image as individual molecule. That means the single molecule. So that's how the single molecule microscopy setup is required or need to develop to observe this working of cargo protein on the microtubules. So again, another problem. So we need to develop them. We need to improve the resolution. That's the other question. So now, after the discovery of the super resolution in microscopy, I can give you a glance how protein normally work on the microtubules. So that's the anchor protein, and those proteins are carrying the cargo and through the microtubules. So they are basically working on that and using the ATP system. So that was discovered later, and 2013 Nobel Prize was awarded for this vesicle trap. Well, so we need to develop a single molecule microscope which can image the single molecule. Before going to that, I need to explain a few of the terms. Okay, a few of the terms. Okay, so you are familiar with the term, the resolutions, right? Resolution. What is resolution? Resolution is nothing. It's a shortest distance you can separate two different spot using your existing optical setup. Okay. So the shortest distance between 2 points on a specimen that can be distinguished by the observer or camera system, and you can separate the entities. Well, so let's say those are the emitters. Emitters mean they can emit the light, and those are you want to observe those points. Okay. So with the existing setup, you can separate those 2 points, but you cannot separate this one. Okay. So your resolution is not enough to separate this unresolvable point or closed points. How to improve the resolution of your microscope? Because we want to observe in a single molecular level. Okay. So the resolution D depends upon the lambda by 2Na. So lambda is the imaging wavelength and Na is the numerical aperture. So what is numerical aperture? So numerical aperture is basically the opening of the objective. Already I have explained you that's the objective of the microscope, which is very important or the most important part of the microscope. And basically it's a incident angle of your light. So if you increase the incident angle, you can resolve the structure. So because Na is in the denominator, right? So if you increase the value of Na, the distance will be shorter. Let's see how it works. Well, I'm increasing the angle and the point spread function or the object is getting narrower. You can decipher each of the individual points. But how much? Also, you have another factor, lambda. Okay. So in 1873, Ernst Abbe, he discovered a law that made the scientists so sad, because he showed that even if you have a multiple combination of lenses in your objective, or you can use multiple combination of lens to magnify your object, but you cannot resolve the point which is closer than the 200 nanometer. How that is? Why he has developed like he said like that? Okay, lambda. Let's say lambda. Consider lambda as 500 nanometer, and place the value here. And your Na is numerical aperture is 1, and because you cannot increase the area much more, that will blaze your sample, that will burn your sample. Okay, so with the existing setup, what you can do, you can observe only 250 nanometer distance. You can separate the entities which are 250, separated by 250 nanometer. But the most of the biological samples are smaller than that. They have the dimension, the virus have the dimension of 100 nanometer, protein 10 nanometer, and even the single molecule has dimension, right? Or it's like 1 nanometer to 2 nanometer. So now biologists are in great trouble. So because you cannot improve your resolutions of the microscope by improving the lenses. So you have no freedom on that. Well, then some physicists suggest, okay, we can reduce the lambda, because if we reduce the lambda, then again D will reduce. Then they try to use an electron beam to excite the sample, and electron beam has much more less wavelength as compared to the normal photon light. I mean the light. So electronic wavelength, as electron gun can purge the electron beam, which can excite the sample, and which has very less in the Armstrong order, it may be in the Armstrong order uh, wavelength, then you can resolve the structure even in 1 nanometer. But the problem is, any sample, if you hit with an electron gun, the electron will hit the sample and it will generate a lots of it, and which will eventually burn the sample, right? So far, people normally use electron beam to image the sample. That's called the electron microscopy. But in the freezing condition, you have to freeze the sample in liquid nitrogen. So you have to freeze the sample. You have to protect the sample with a cold chamber. Then you can apply the electron beam there in electron microscope. But what's the problem? You cannot study the living specimen. You will miss the real-time important biological phenomena, because you want to study the biological system in more realistic condition. Also, as it's a transmitted image, you cannot tag different component of the cells or biological specimen with different color. that will give, so electron microscope will give you a black and white. So you cannot detect them. Okay. Black and white in the sense you cannot use multiple colors. Okay. So that's another problem. So we need to develop a technique which can break the diffraction limit of 250 nanometer. Also, we want to image it in the realistic condition in the lives. Okay. Well, so can you imagine what is that? That's a galaxy full of stars, and that image has been captured using a Hubble Space Telescope by NASA. So why I'm mentioning that, I will explain. Okay, so you want to observe single molecule under your microscope, right? And it's basically same. It's a night sky under your microscope with full of stars. Right? Here, they normally observe multiple emitters. Emitters mean the point source which can emit light. Okay, so there are multiple stars, or maybe not star, maybe some different objects or different celestial bodies are there. So their job is to identify each of them, identify their location, and then they can track their movement. That was the task. So how astrophysicists do that? Okay, so any point emitter will create an impression on your detector. How that is? So it will be like that. It's a spread function. So we call it a point spread function. So you don't need to remember those terms because that's not mandatory. But just imagine you have a point source and you try to I try to fall it. You try to fall it on the detector, and you will get an impression. How it will be? It will be like that, and that is a large impression. What is the exact location? That's the main question. So your star will appear as this large spot, but you have to locate the exact position. How you will do that? Let's draw a line through the impression. What you will get? Okay, if I want to plot the intensity versus pixels, what or position, what you will get? Eventually it's very less intense, and gradually it's increasing, intensity increasing, and then again it's decreasing. And we can image, or we can get a impression, same impression on the other axis, and you will get a three-dimensional Gaussian function. And you can easily feed this type of equation by that's a 2D Gaussian. You can by using this mathematical equation. Oh, yes, now chemistry physics has the collaboration, nice collaboration, and also the mathematician help them to resolve the structure. So anyhow, if we can resolve the center of the spot, that will be the exact location. Okay, so that's how scientists locate the stars. The astrophysicists locate the stars. But the problem is that if you have less number of photon, let's say n, then how it will appear? So it will appear as a flat curve. Okay, and it is very difficult to determine the center of a flat curve. That's why the good emitter, that means which can provide the maximum number of photon, that is a good emitter. You can precisely locate them. That's the basic thing. Okay, clear. Now, can we use this approach in the super resolution microscopy? Yes, we can approach. So our task is to just localize those two spots separately, because those are not resolvable. These two spots are not resolvable by using our existing fluorescence microscopic setup. I want to resolve them. So what will be our approach? If anyhow we can switch off one of the emitter, then we can image it separately, and for the next frame we can switch off this one and image this another point. So you can separately image them if they switch off and switch on accordingly with respect to time. So we don't want to get all the emitter switch on at a time. We want to switch off and switch on with respect to time. So people has developed called blinking dye. They can easily switch on and switch off their fluorescence with respect to time by changing their electronic state. Most of okay. So I am not going into the details of the electronic state back because that may be difficult for you. So let's just have a look on this type of dye. That's a blinking dye where those type of dye is normally switch on and switch off. Okay, how it look like? Let's say. Well, so probably you have to switch off the light of your room to get a clear view because so can you observe it? Is it visible, Sadin?
Yes, yeah, yes, it's visible.
Okay. Well, so now we are using a different type of dye which can blink, and I will switch on it and switch off it, and eventually how we'll determine the position? Well, anytime you will get this type of impression, that's called the PSF, and if you fit it and resolve it, then you will get this type of localization. Now I can switch off one part of this PSF, again switch on it, and then I will get multiple point, and if I construct or gather all the multiple points, then I will get a super resolution image. I think it's clear for now, but I will explain in details later. Okay, so how it improves the localization? Let's see. So this is the nuclear pore, which is the main device which allow the signals to transfer to come into the nuclear environment, and or let's say it's a machinery which is used by the nucleus to transport signals from the cytoplasm to nucleus. Okay, so initially it was a average picture where you cannot detect the pore, but using the super resolution microscopy you can easily detect them. So initially it was 1.2 micron, now you can resolve even 50 nanometer. So yes, we can break the resolution or break the limit. I mean, it was initially 250, now we can image even 50 nanometer. So that was the previous impression or image of the actin filaments with the standard fluorescence microscope, and now with the super resolution microscope you will get this image. That's a image of mouse kidney. Initially it was bit blurry. Now with the super resolution microscope you can get this type of. Well, those blinking phenomena helped us to get that, to break the resolution below the 250 nanometer. Okay, that's why in 2014 the Nobel Prize was awarded to Bedzik and Moriner and Stephen Hale for the discovery of this technique. Okay. Well, so now I will explain what we are interested in, or I am interested in. So people are aware about the mechanism that how the cargo exported and imported within the cytoplasm, but each and individual organelles have their own function, right? And nucleus is the head office of that because it's contains mostly it contains the genetic material. So it is very important to decipher whether or how they can export and import their signal to the cytoplasm. So that was our motto to study, or that was our basic question, that how nucleus crosstalk with the cytoplasm. Well, initially it is discovered that how cells crosstalk between the organelles using the microtubules, as I already discussed, but it is still not clear how the nucleus crosstalk with the cytoplasm. To decipher that, we have to identify the machinery through which nucleus exchange their signal. That's called the nuclear pore complex. So that was imaged. So this why that? So you can ask me, how do you know that that structure will be like that? Yes, you can image it using a electron microscope, but in a freezing condition, it looks like that. So it has two rings. One ring is on the cytoplasmic side, another ring is on the nucleoplasmic side. Okay, and those particles need to be transported or imported into the nucleus, or some particles should be exported from the nucleus. Well, we need to study those tracks. That's our prime goal. So what is the basic structure of the nuclear pore? That structure has been obtained from electron microscope. So basically, it consists of different proteins, 30 different nucleoprotein proteins. Some of the protein construct the or form the two base rings, which is octahedral, which has the octahedral symmetry. One is on the cytoplasmic side, another is on the nuclear side, or the side of the nucleus, and some protein which looks like a filament, they generate a selective net. Normally the cargoes or the signals, or let's say some molecules which is less than 4 nanometer, they can easily pass through this channel. But more, the particle which has the dimension more than that, that means it's maybe, let's say, 100 nanometer or 50 nanometer, it will not allow those particle, or it will selectively allow those particle to enter into the nucleus. Well, so now the question is, how the cargoes which is greater than 4 nanometer transported through the, how are the cargoes transported through the NPC? NPC is the nuclear pore complex. So it's a short form of the nuclear pore complex. Okay, I'll use the term NPC. Okay, so now our question is, so that is the main tunnel, and you have to pass several cars, or you have to transport several cars with different size with this tunnel. So what's the main problem? Why we are studying that? So those car has to pass through that channel within millisecond. So millisecond mean 10 to the power minus 3 second. So lots of the molecule are coming out from the nucleus and going into the nucleus at same time. So there is a possibilities of traffic jam, right? Because tunnel is very narrow. It's in the order of 60 nanometer. But your molecule itself has different size, and it has to transport it. It has to be exported, imported. So anyhow, there is a possibilities of traffic jam. And if traffic jam occurs, what will be done? What will happen? So most of the car will clog there. That means the protein will clog there, and it will create a different type of disease. Okay. So what is those? So normally, if you are not able to transport, or if the protein is not efficiently transported into the nucleus, or it cannot be exported or imported through this channel, they try to accumulate there, and there is a traffic jam, and there is a possibilities of aggregation of the protein, and those aggregation cause severe effect on the health. So what was those disease? So probably you are not familiar with the term. It's ALS, leukemia, and Alzheimer's, Huntington. uh, I can mention one example. So uh, last year uh, we have lost one of our favorite scientists, Professor Stephen Hawkins. So he was suffering from ALS. It's a disease due to the protein aggregation. Okay, so some genetic disease, or some disease which is normally now very usual in the late age. You cannot solve it. So you need to know the tracks first. Well, so our task is to decipher the tracks. So initially we try to decipher the track using a single molecule. So this molecule we have, we are able to track this molecule using our existing microscope. Probably you can you can observe it behind me. It's sitting behind me. So this molecule is trying to penetrate the nucleus barrier. Nuclear barrier. This is a one molecule. This is just one molecule. That's a single molecule, and this molecule is trying to come out from the nucleus. That's a abortive event. Okay, but we can have we can feel that this molecule is trying to enter or trying to get imported or exported from the nucleus. But how? So it's basically a pixel, right? If you zoom it, if you magnify it, you will get some pixels, and these are the cargos. We have tagged it with different color, and those are the nuclear membrane. So this is your car, and the green color is your tunnel. But where is the tunnel? Where is the car? That's just a pixel, because most of the pixels are more than 240 nanometer. So you can ask me, we don't know the exact location of the pore, and even we are not able to observe the pore. So you cannot make us fool. Well, I will not make you fool. So that's our main task, to get a 3D structure of the real pore. Till now, with the existing technology, how people image a 3D structure? So people can image each of the individual plane with different time, with respect to different time. Let's say you can image this plane and again image this plane and again image this plane, and if you combine them, it will give you a whole structure or 3D structure of the sphere. But if I ask you, well, when you are imaging this spot or this plane, what is happening at that plane? That means at a particular time, you cannot probe all the plane. If you cannot probe that, then you are missing an important parameter, that is time. So your imaging system is not that much efficient, because you are not able to observe the 3D, you are not able to construct the 3D image with respect to time. Why that is? Because if you are trying to focus a plane, let's say z equal to 0, and if you are above the plane and below the plane, the impression of the image will be same, or it's kind of similar. Okay, so you cannot detect the image, you cannot detect the specimen or phenomena which are happening below the focus or above the focus. So far you want to decipher these tracks, you need to develop a technique so that those spots can be appear as separate entity or with the different shape. So what we normally we use, okay, so I think you are familiar with those pictures, right? In the rainy season, normally that was the so if the weather is clear, then you can observe the view like that, and sometimes those light appear as an elongated impression in your eyes, because due to the change in the refractive index in the weather, or due to the problem in your eye, normally the impression in the x and y will focus at different points in the detector of your eye. So that means that's called the astigmatic view. And if you are facing this problem frequently, I think you need to consult the doctor, because they will suggest you to use a cylindrical lens, which will eventually correct correct your view, so that you can get a uniform image, so that this focus can be adjusted in a same focal plane. What we have done, we have used the reverse way. Okay, we have used the cylindrical lens, but in an opposite way. we want to observe the PSF to be elongated if they are above the plane or below the plane. How that is? Okay, if you change the focus, that will appear as a line. Now it's again in the center, and if you are above the focus, then it will appear as a line, but it's extended to the another direction. So that concept has been developed by using a cylindrical lens, but we are not happy with that. So because you can generate a different particular type of pattern with that. So like that, you can generate a particular pattern, but not all pattern which you want. That means, let's say I want to generate the PSF, I want to decipher the structure which is which is just below the 50 nanometer or above the 50 nanometer. Then you need to develop a more elongated version of the PSF. So we use the existing technology that is used for the in the Hubble's telescope to decipher the exact location of the stars which are normally giving you some elongated view. So already I have discussed that there are some stars or infinitesimally small object which can emit light in an elongated view. So how do you locate their position? So people normally use the combination of mirror which can deform with respect to voltage, and you can generate those. Okay, I am not going into the details because that's the electronic part. So now you can have an impression, you can determine the G position at a particular time from the astigmatic PSF or from the shape of the PSF. And if you have a calibration curve at a particular time, if some particle appears like that, then you can determine their G information in a real time. Well, now we can determine the G information. Okay. So our next task is to decipher the structure of the nuclear pore. Yes, we have selected those octahedral protein and we tagged a blinking dye with the protein. And now in 3D, initially those spot was round, as I already discussed before. So now the spot are astigmatic. So some spot are perfectly, it's in the diamond shape, and some of are elongated. So elongated spot mean they are below the focus, they have different G height, and above the plane they is elongated to the y-axis. Okay, and now we can easily construct a super resolution image. So what is the basic concept? You have blinking dye on your pore. So already we have tagged a blinking dye, and you can reconstruct the image, and yes, you can feed those points, and what we are getting now, we are able to record, or we are able to develop the 3D structure of individual pore. So it's the individual pore in real time. So you don't need to freeze it. It's imaged using our existing technique which we have developed, and that can image the live cell. Okay, so that's the nuclear pore of a live cell, and you will get eightfold symmetry. Well, yes, so we have multiple dye on that, so that we can track the cargoes. So what we have done, so we have first tried that, okay, we have to try whether our car are at all viable to penetrate the nucleus. Well, we have applied some proteins which should be transported into the nucleus, and yes, they are transported because they are, with respect to time, they are gathering within the nucleus. So that is acquired with the using a normal transmitted light microscope. And with the super resolution microscope, with 3D super resolution microscope, yes, that is an individual protein, and that is trying to penetrate the nuclear barrier. And where is the pore? Yes, that is the pore. And well, so the red color is the impression of your pore or image of your pore. That means the two rings, and those blue color, those green colors are basically the track. So what people normally thought about the nuclear pore. So their pre-existing hypothesis was that normally the cargoes are they normally try to use the central channel to get exported or imported. But what we observe is that no, so even they can use the peripheral channel. You can observe that there is no such track exactly in the middle. Although that's a very preliminary stage, so only 160 localization is there. So we have to wait because we are trying to gather more point, at least 2000. Then we can emphasize on that point, and we can claim that okay, well, the most of the cargoes are trying to use the peripheral channel rather than the central channel. So that's in the real time. Well, so now you, you can image any biological sample in 3D, which is in the real time, which is in living condition with the resolution of 10 to 15 nanometer in X and Y and 20 nanometer in Z. Well, yes, that's the summary. So what we learn is that it's equally important to study physics, chemistry, biology, also the mathematics to develop a new technique. So normally in school, I don't know what I am not now I'm not aware about the syllabus which is now people are using to study. I mean, what is the present syllabus? I don't know, but in most of that cases, it happens like that. Okay, I will read selective. So chemistry is my favorite subject. Physics is my favorite subject. That maybe, but you need to learn everything to develop a new technique. Okay, so I'm not going into the details. It's already 1 hour. Yes, that's the microscope. It's sitting behind me, and those are the important parts. So as I already told you, that is the eyepiece, and this is the stage where we place the sample, and we have two cameras. One is for to image the sample in a fluorescence mode or confocal mode, and another for the super resolution. And that's the deformable mirror, and those are the optics. And yes, I'm really thankful to Professor Siegfried Moser. He's the PI, and also I'm really thankful to ma'am, Professor Sumita Basu, and she has joined here, and I'm really thankful to him. And I'm also thankful to Rajdeep, Dr. Rajdeep Chaudhary, and Sandeep Dave, and Ankit Sharma. And obviously, I need to thank NIH for giving us a sufficient amount of funding. And thank you for your patience. And my talk is open for the discussion.
Okay. So,
Well, I need to show you something. So students are there, right?
yes, yes.
Okay, can I change?
No, you can just
Stop
show.
sharing, right?
Yeah, stop sharing.
Stop sharing.
Yes.
Well, so I need to show you the example of fluorescence. Okay, so this is Alexa 568 solution. That means it's one dye. And why I have already told you that it can give us back a light with different color. I will explain it. Okay, so this is one dye, solution of one dye, and this is the laser. Okay, so I can place the, so I can you see the different color? Yes. So initially the color is different, but when I excite the sample with a green laser, it will give you a nice fluorescence. Using, even it can give you different color if you excite with different excitation source and with the different molecule. So that's the fluorescence basically. Okay, and I can show you the existing microscope setup.
You can kind of use your smartphone now. You can
Yeah.
you can switch off the video and the microphone in this thing. Yeah.
Okay. It's a feedback, right? Well, let me rotate the camera. Okay.
And now it's fine.
Yes. So that's the body of the microscope. So it's the eyepiece, and this is the white light source, and those are the lasers. Okay. Let me open it. So those are the lasers to excite the sample to get the if you want to get the fluorescence image, and those are the optics just to focus the laser and into the sample. Okay, and these are called EMCCD camera, and you can image. It's a very sensitive camera. Even you can image the samples in the millisecond order. Let's say you have time resolution of few millisecond, and so any phenomena you can capture it, which is occurring in that time resolution, that means the millisecond order, and that's the deformable mirror. That's very sensitive to light. That's why I have covered it with the black color, black text and black cover. And that's another camera. Yes, and this camera is connected to the computer, and that's the basic setup. Okay, so well, if you have any question.
Yeah, so you can kind of go to your main laptop thing. You can sit there, and I can put up.
Sajid, can you repeat it? And I can.
Yeah. Okay, that's fine.
Okay.
You just switch on. Okay. So there are very few questions. The one of the issue I kind of asked, like there is not many people from biology background, and that did not kind of follow some of the stuff. But still, there are like at least two questions from the students. I'm just pointing out. So
Yeah.
from the Abbe equation, correct? You showed. So
Yeah.
there one Ronak Kumar Das is asking how to calculate Na and what is Na.
Okay. So let me share the screen. That will be better. Okay, share. Yes. So Na is basically the angle of your incident light. Okay. So you can, so let's say you have a spot. Okay. And you can change the divergence of your light using a combination of lens, and you can measure that angle, because we have that setup and you can easily develop that. It's nothing, just to measure the divergence of the, how much divergent is that and where it's getting focused. So using this technique, you can observe your spot and you can measure the angle. Depending on the angle, you can measure the Na. It's basically depends upon the opening of the objective. Okay. And yeah, I can send, I can explain.
So then this is not a fundamental limit, correct? So you can kind of change Na if you change the lens, change the opening, if I understand correctly.
No, you cannot, because, okay, why you cannot change more than that? So your limit is 180 degree, right?
Mm-hmm.
So n sine theta,
Mm-hmm.
what will be the value?
N sine theta, like what is n? N again is the number, correct? So that you can control, no?
That you can control. Okay. So, but that is basically an angle. So if you change the angle, you can vary the angle up to 180 degree, not more than that. another problem is that that light may create a total internal reflection. So even you increase the area, you cannot cannot control this more, you cannot control this angle more than 180 degree. That's the limitation. That's why even if you make the area larger, you cannot go beyond, let's say, 2. That's why most of the most of the microscope which is extremely sensitive, they have the Na around 1.7, not more than that.
Mm-hmm. Okay, okay. So, ask
Well,
another question from the students. So there
okay.
is some blinking related question. So how do the blinking dyes work? Is the blinking essentially random or can it be manipulated?
Yes.
And what is
Yes.
blinking dye? Yeah.
Okay. That's a good question. Okay. So those blinking phenomena is stochastic. That means it's random. Okay. So, okay, let me again play the movie. So follow the square box. So it's the same position, but multiple time it's switch on and switch off. How those does dye change their electronic state or the blinking phenomena or their fluorescent on and off system using by changing their electronic state. Okay, let's zoom it. So if your dye will emit the photon uh, from the singlet state, that will be the fluorescence, right? But anyhow, you can devise or frame the dye like that, you can synthesize the dye like that, it can easily switch on and switch off between them by changing their electronic state from singlet to triplet. Then you can make them off at a particular time. And some dyes, they are activatable. So let's say this dye is sensitive to 450 nanometer, or let's say 500 nanometer. If you apply another line of light or another laser line, that means another light of in the UV range, so normally it reduce their photoactivable state and it can switch off the dye. And by changing this activation, you can photo switch the light. Okay. So these are basically rhodamine based dye, and yeah, it's very difficult to develop those dye because now there are only, let's say, 10 to 20 dyes are available in the world to image to use for the super resolution microscope, to use for the super resolution microscope. So now the job of the chemist is to develop multiple dye with different color which can blink. Yeah, that's a challenge. So that's the way they just normally they change their electronic state with respect to time, then they are in on state and off state eventually. Okay.
So another question is that can you observe the original color of cell organelles?
Well, the thing is that you can observe, but the problem is the intensity. So using a white light, you can observe the magnified view of the cells or the organelles, but you can separate them because if your light is not viable to penetrate the uh, cell in different slice with respect to different z height, you will always get the negative impression like that. I mean, the negative image like that. That's the thing. Well, let me show you. Yes, this one. Okay, so with the white light, that's the average structure of the cell. But to observe the organelle, because the okay, the more important point is the organelles are in the diameter of, let's say, few micron, 1 micron to 2 micron maximum. They are, and some of them are like 500 nanometers. So how you will observe them? So you cannot observe them by by observing with the transmitted light microscope. You need to use a fluorescent light because the cell is a total factory. You have to separate the entities from that, right?
Okay, so another like just a curious question from Shuham is that like when you are using electron microscope on neurons or like maybe small cells, don't they
Mhm.
generate unnecessary impulses, and for that heat it destroys the cells or that?
Right. So the problem, okay. So basic, the fundamental concept behind using the electron microscope is to reduce the lambda. Okay, because electron has a wavelength in the Armstrong order. So then you will get the lower resolution. I mean the higher resolution. That means the lower D. But the problem is that electron has a wave particle nature, right? So any particle that will hit charged particle, basically, it's a charged particle. First of all, it can change the charge character of your biological sample, let's say protein, or any charged particle. Any charged particle, if you try to heat the charge those specimen with a charged particle, it will generate a massive heat and also generate a charge. So to prevent that, people normally snap freeze it. That's why electron microscopy is very efficient to decipher the structure in the Armstrong order, but for the dead sample, not for the live sample. Okay.
Okay, okay. So another question is that can you use this technology to study microplasma as well?
Uh, well, yeah, you can, but still, the problem is the selective labeling. So you need to have a selective label for that. That means the selective dye for that. So till it's not developed yet, as far I know, so it's a lack of proper dye or proper label. After that, we can use.
Okay. Um, so another question is that why are you there, Abhishek?
Yeah, 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 yeah.
Okay, why all fluorescent have less protein counts?
Less photon
Protein
count.
counts, yes.
Okay, so why most of the fluorescent protein are less fluorescent, right? That's the question. If I
Yes, yes.
am correct. Okay. Okay. So why those protein are fluorescent? So they have a fluorescent moiety. Okay, that's called chromophore. And those chromophores are extremely sensitive to the environment. Okay. So it depends upon the environment is hydrophobic or hydrophilic. That means if you have a hydrophobic environment, the fluorescent peak will be in another position. If you have hydrophilic environment, they will give you a different. They will give you the fluorescent with different color. So it's very important on the packing of the protein. If those fluorescence moieties are exposed into the solution, then you will not get any fluorescence or you will get faint fluorescence. So when you irradiate light, light also generate heat and the structure get ruptured. And as the structure get ruptured, then the fluorophore get exposed to the hydrophilic environment and they are not viable to generate uh, the fluorescence in the visible range. That's why it's easily get degrade. And other thing is that fluorescent protein are attached with other protein or maybe other organelles. So if you blaze it with the light, then you can also there is a possibility is that you can damage the link up. So sometimes fluorescent protein get aggregated there, and you will get a blob, not the individual fluorescent
Okay,
protein. Okay?
okay,
That's another problem. That's
okay.
why the
So,
fluorescent
yeah,
protein are not that much efficient for longer time scale imaging.
yeah. So there is an interesting question. I also had this in mind. Like people are using electron, correct?
Right.
Electron microscope. So what I'm asking, like, can quantum mechanical effects or like quantum particles to have a
Right.
quantum microscope, correct? To get more smaller quarks or something. So one is quantum mechanical effects or some quantum particles can be used for microscopy.
Quantum, what do you mean by quantum particle? I mean, uh,
So it's like, so
so
there is like electrons are
which
like,
of the
so
energy state
electrons
is
are
quantum?
consisting of multiple quarks. Assume that, correct. So then,
Okay,
so can you use that or use some quantum mechanical effects? That's another separate question. Quantum mechanical effects.
okay. Well, so the first problem is that so if so energy and time, those are commutative. Okay, they are in a commutative relationship. So if you have energy parameter, then you have to compromise with the time. Okay, so any phenomena which is not quantized, you cannot use them for this imaging purpose because it's totally depend upon the quantization process. You have to have a definite energy gap. You have to have a definite wavelength from for your particular dye. So you can call it as a quantum phenomena, but I'm sure what you are exactly trying to develop. If you can explain me in details, I mean, what is the basic idea? So you want to use a excitation source, which is something else, quartz or something else. What is that? I mean, if you
So
clarify.
like, for example, like why are you using electron in the first place?
Yeah, to get the wavelength, smaller wavelength as compared
Yeah.
to the light.
So is there like that? Is that the limit of smaller wavelength?
No. So
So
till now, okay, I got it. Okay. Well, to generate, so always you have to think of the generation of the electron beam or light source is feasible in the existing condition or existing setup or not. Yes, you can do that, but to generate those type of particle, it's a highly heat consuming, right? You need to develop a high temperature chamber, and you need to develop a setup which has which will require others, which will require a completely different setup and another room to generate the gun or to generate the
yes.
beam. So yes, now
So
people are able to observe in the Armstrong order. Let's say now the limit is 2.8 Armstrong, or let's around 2 Armstrong using an electron microscope. So people are happy with that. So I mean, you can develop if it's required. Even people can image individual molecule. You can image the each of the carbon ring of the benzene, carbon molecule of the atom of the benzene. Okay, so you can
okay.
even image the whole six-member ring.
So there is a student named Saraswat Sen Sharma. You can ask directly your question. So
Yeah,
you can
yeah, yeah.
unmute.
I want to hear from
You can
them.
you can unmute and ask directly.
Yes, sir. Thank you, sir. Sir, my question was that since this is a very small system, is it possible that just by observing the system, we are changing how it is actually behaving? That is, maybe a biological process turns out some other way, just because we are observing it.
Mm, okay. Okay. So can you repeat the question again? So it's not clear. So
So
what?
what he is saying that because you are observing, correct? So
Right.
are, and you are kind of putting fluorescence and some electron, like something, correct? So are you not essentially changing the biological process itself? So that like a biological process getting changed because of the
Yes.
observing.
That is very important phenomenon. Okay. Okay. So your question is like that: whether you are perturbing the actual phenomena when you are using the light for the excitation, right?
Yes, sir.
Yes. For that, so what we do, we always check whether the low power and high power affect the process. So that's why you have to perform lots of control experiment where that the cell is viable to the light at that particular wavelength, or how much time they are in living condition under the microscope. So that you have to calibrate. Yes, it will be perturbed, but that's why we try to image the sample in more realistic condition. Without perturbing anything, you cannot image.
But
Even
how do you define that realistically?
even you observe any object, you have to observe with the light, and light is interacting on the object. So that is basically you are perturbing the sample, or you are perturbing the object. So yes, that will part of the biological process, but in a minimalistic way. Okay. So we that's why we perform the lots of control experiments, so that the biological sample we can mimic the realistic condition of the biological molecule. Clear?
Thank you, sir. And sir, just one more question.
Yeah.
Because let us think about the double slit experiment, Young's double slit experiment. In that experiment, which slit the electron goes through is actually not important and essentially undefined until we observe it. So,
Yeah.
in your research, the part that you say that whether through the NPC complex it is going through the central hole or through the peripheral hole, is it important or is it even defined?
So
That is
that's our
because we
job
are
to define whether,
small.
because yeah. So the thing is that it is very important. Why? Because the peripheral channel is very narrow. So it's of 10 nanometer, and the central channel is 60 nanometer. So if you delivered a protein or the cargo which has the dimension more than 60 nanometer, and still they are trying to use the peripheral channel, that means there is a possibilities of not transporting that protein into the nuclear environment. So that's why which cargoes or which protein follow which path is very important to decipher. Okay.
Shubham, you can also ask your question orally, and if you have anyone has any question, you can directly
Yeah,
ask
yeah, I
orally.
love to answer that, and I want to hear from you.
Shubham, your question you can ask directly orally.
Okay, thanks a lot, Dada. So, particularly, I am having currently two questions. First one might be a dreamy one. So, like using this fluorescence protein, can we bioengineer a certain plant species that can actually give out some light during the night just to use
Yeah,
that as a night lamp or something like that?
good. It's not a dream. It's
Might be
already
a dreamy.
yeah, no, it's not dream. So obviously you have to dream to develop something, and now people are trying to develop the transgenic plant which can give you the light. But the problem is that those fluorescent. So if you so the other process are there, no. So you have to maintain the other biological process to make the make the tree in living condition. So people have already developed it, but the problem is that those are not um, they are not in living condition, let's say for more than 15 days or 10 days. So
So,
it's not.
are people using CRISPR techniques to kind of cut and paste some fluorescent thing on the thing?
Yeah,
Is
that
that
CRISPR technique is it's basically editing the gene. So you can label as per your requirement. Let's say I want to label the protein in a particular site. So I will select the cysteine. I want to place my cysteine residue there. Then only I can tag the my particular dye, right? So I will do the cut and paste, and I will paste a cysteine there, and then I will tag the sample and tag the biomolecules. Well, so your answer. So answer is that yes, you can do, but also you have to maintain the other condition. If you can, people are trying to develop that. Then like bioluminescence, right? The fireflies provides the bioluminescence. So you can develop the transgenic plant. Yes. Then your next question.
But why does it have such a short lifespan?
Because so if you filled up the whole cells with the heavy concentration or heavily concentrated fluorescent protein, then the other protein are trying to they are not behaving uh, regularly. So in the sense they are getting aggregated, so other biological process are getting hampered, and then even those fluorescent protein are normally they try to kill some organelles. So that's even the main problem is that the tree has, or the transgenic plant has to perform the photosynthesis, right, for their living process. And so, but they are not anyhow not able to develop the chloroform, sorry, the chloroplast into their microenvironment, so into their cells. So that's why they are not right. They are not able to they able to live for 15 days or more than that. That's the problem. Yeah, yes, people are trying to develop, improve the condition. Maybe after a few years you can develop that. It's not a dream at all.
Yeah. If anyone has any more question, can
Okay,
ask
that
directly.
was quite nice to know. Okay,
Yeah.
so my second question is regarding quantum biology. So recently I just heard about this field where you use quantum mechanics and those like probabilistic curves and all to actually detect and find the actual procedure of how the photosynthesis is happening at the fundamental level in chlorophylls, in the chlorophyll pigment. Okay. So regarding that, I was just asking, since light has to travel through all the possible paths it has, but the probability shifts to the one particular path, doesn't, like it's related to the question Sasato asked, while we are actually measuring the blinking pigment, the blinking fluorescence. So doesn't it kind of like gives us any false feedback or something like that?
No, it's totally different. Okay, so for quantum biology, so if you want to study that, so why normally light is required for the photosynthesis process, synthetic process, synthesis process, because light will induce a electron transfer reaction, right? So it will goes to multiple electron oxidation reduction process, and ultimately it will give you the water and the oxygen. That is different. But here you want to image a sample which is less than 50 nanometer or which is closer than 50 nanometer, and you want to decipher their exact position. So if they are on at a particular time, then you cannot image them, because both will blaze and both will give you a average peak, or average or a soldered peak, let's say soldered peak, like the Gaussian. So you cannot resolve their position if they are at a time they blaze. So one has to go off. That's why the blinking phenomena occurs. So the main motto of the blinking is to make the molecule in the off state most of the time. Very few molecule will be on, and after some time, or after next frame, let's say after, when you image one frame and when you are trying to image the other frame, then other molecule will be off, and which was off, that will be on. That's the basic idea. It's not related to the electron transfer. Okay. So am I clear, or I need to explain it again?
I think you can kind of do it offline.
Can you just, if possible, if possible, that can you elaborate?
Okay. Okay. So can you? You can switch on my video, right? Okay.
So I think.
Okay. You want to observe. You want to separate those two fingers in a super resolution image. Let's say it's a microscopic object, and those are separated by, let's say, 10 nanometer or 15 nanometer. And so what you will do, you will label the finger with dye, as I already told you. So you will label the normal dye, and when you apply the light, it will excite the dye on the finger, and all the finger will give you the impression like a average image, like, right? So you will, because they are blazing, so you will get an impression, but not the separate one. So to get the separate one, or to construct the separate image, what you have to do, you have to either switch off one portion when you are imaging, and then you switch off the other portion when you image the next frame. So first thing is that, let's say I'm imaging in 1 second. So that portion is off. So you can image that. Again, in 2 seconds, I can switch off this one. Then you can image this one. So you need to continuously switch off and switch on the individual dye. And by fitting the Gaussian function, you can construct the image of individual finger. So let's say you have to record 10,000 movies to construct the image, because you have to switch off and switch on individual dyes. That's why it's called reconstruction image. It's a STORM. So the technique is called STORM, stochastic optical reconstruction technique. Okay, or microscopy. Now it is clear?
Okay, I think it's like
Yes, yes.
partially clear. So, so anyone has any
Thanks a lot.
question directly? Anyone has questions directly? You can ask.
Sir, I have a
Yes, sir.
question.
Yes.
Ask.
Sir, is there a limit to how how many lumens bioluminescence can produce? Like
I mean,
brightness can be as much high as we want to.
okay, yeah, no, that's a good question. Yeah, every dye or individual dye has a limit. Even you increase the power, it cannot give you the maximum. It has a limit of maximum number of photon it can provide you. If even you increase the laser power, it will not provide more than that. Yes, that has a limit. That's why there is a limitation of photon, and we normally try to use those dyes which has the higher limit. I mean, it can provide you the maximum number of photon. So the good dye can provide you the number of photon, more number of photon, and the bad dye, they have the lower limit. I mean, they can provide, even you increase the power, they have the lower limit of photon.
Okay, sir.
That's a
Thank you.
good point. Hmm.
Anyone can ask directly if you have anything.
Sir, I actually had another question, a mild curiosity, that for taking one image, how much time is it required? Because since you're switching on and off so many times, it should take a long time.
Yeah, so okay. So normally to construct the image of nuclear pore, you need to record 10,000 frames. Okay, and each frame is of 10 millisecond. So how much it will take?
Then it takes 1,000 seconds.
So 10 millisecond into 10,000 frame.
Yes.
Millisecond into 10,000.
100 seconds. 100 seconds.
It's 100 second? No.
10,000. So 10 to the minus 3.
1,000 second around that, right? 10 to the power minus 3?
10 to the
Well,
10 cube. Yeah.
that means how many minutes?
So more than 1 minute. I mean, about 2 minutes.
Yeah, it's about 2 to 3 minutes. So in 3 minutes, you can image 10,000 frames. That's not,
Yes.
yeah, impossible. So you can easily image. Yeah, the most of the important thing is you have to acquire a high amount of data. I mean, let's say to develop one image, you have to gather, let's say 3 GB data for one pore. And the cell has itself, let's say, we can image at a time 50 pores. So you need to have that much capacity of your computer, because you can analyze each of the individual frames. So computation is required. So yeah, Sadin, I need to thank the engineers for this computer engineers for developing such a nice program.
Yeah, so there are so many engineering things. Yeah.
You can analyze the image. Yes, sometimes you have to write the program from your own self as per your requirement. But yeah, the existing program are there.
Yeah, even the like lens design and everything, there is so much engineering aspect of every aspect
Yeah.
has engineering. So you can't, as you said, physics, chemistry, mathematics, and you can kind of separate the engineering aspect.
Yeah, engineering is basically combination of physics, chemistry, mathematics.
Yeah, so basically you are solving problem, correct? So
Yeah.
we have just giving some names. So anyone has any questions? Not like I have a philosophical question, correct? So you said that every subject is like every problem is you have to understand so many stuff. So why do we have subject boundaries now, correct? So you should study problems, correct? And then whatever thing happen, you learn from that, because we name it. Then what will happen? Like we learn physics part of it, chemistry part of it, maths part of it, and you kind of specialize in some component. So should we not like interest in like understand the problems in general? Like if I understand microscopy, correct? I used to study microscopy, correct? If I am interested there.
Uh,
So why do we need subject boundaries now?
okay, so so it's a very good, it's a good idea, but I think it's not realistic for now, because so you need to have expertise on a particular subject. That's why it's not. So you need to you have to have a teacher for that. So one teacher cannot teach everything at a particular time, or in a particular school. So some of them are expert in mathematics. So that's why the subject has been compartmentalized. So that some people can teach, who are expert in mathematics, they can teach mathematics. They can, some of them can teach physics. So that's why.
Yeah, but
Let's say
that's a good.
you are
That's
expert in engineering, so that's why it is not possible to acquire all knowledge of chemistry for you. So I need a, if I want to teach that, then you need to have an expert of chemistry or expert of
no, but
engineering.
the problem is like, but the problem is you don't make connections very fast, correct? What
That's the
happens?
problem. Yes, I
So,
agree.
so
So
the thing
most,
is, so the, for example, yeah, we learned kind of calculus, correct? Integration,
right,
correct? But calculus integration to physics, how it is connected, correct? And when you learn like SPDF or the electron thing, how that is connected to the physics, chemistry, maths of it, and you are saying the stochastic, correct? The probability is so like quickly coupled into any physical system, correct? So
right,
now that comes from that, like whenever the initial things discovered because of those motivations, correct? So what
right.
happens, like people don't make those connections, and over like
Yeah,
after doing undergrad, masters, you don't make connection. Then suddenly when you are doing research, you are trying to make connections. So
yeah,
you
that's
are
the
kind of,
problem. So
so yeah.
always you have to you have to try to find out the missing link. So the link is very important. So if you are not able to connect those, it will be a burden for you. Let's say physics, chemistry is separate entity as
But
well.
the problem, if you specialize, how do you connect? Because you're just learning a specific
Now that's
stuff
the
over
job
there.
of the teacher, because I'm really thankful to my ma'am. Probably I don't know ma'am is present here or not. I'm really thankful to ma'am, because when I was in the college level,
But it is too
some
late,
of
bro.
my
You're saying the
teachers,
PhD, correct? So after like school, undergrad, you are
yeah,
not doing that,
yeah,
correct?
in undergrad people can do that. So that depends upon the academic environment. So some of the good teacher helps us to develop to connect those missing link. Some of not. So it's a problem. I agree that it's a problem of teaching process, or maybe the system, because we don't never connect them. So but I am really thankful to some teachers. They, I believe that there are some teachers who are now giving their lecture, um, uh, just to connect the. So
But those are
I
not
think the
process.
school.
You have to be very
The major
lucky,
thing is
correct,
that,
to
okay.
come up with
So
those
the major thing is,
teachers,
I think
correct?
the school need to organize those events so that people can connect everything. So that
Yeah.
class will be on the missing link of physics, chemistry, and mathematics.
Yeah, yeah. So also like when you
Yeah,
are studying
that's the thing
engineering, you
that I
have to
agree.
know mathematics. So everything is connected. Basically, you are solving problem, and from that problem, you have created the subject boundaries, correct? And
Right,
then
right.
and you know what happens, like even for in the research community, like everyone has their own silos, so they are kind of solving same problem in different ways, correct? So then is there any waste of efforts? Like because you are like engineers solving this problem, and physics, chemists are solving the same problem, physics are solving. So what I'm saying that there should be some way, like some processes, correct? You should not depend upon a few teachers. That's a lucky event, correct? So you, like for example, as you said, like there is some courses or something where you
Yeah, yeah,
kind
yeah.
of have
I agree
this
with you.
in the
That's why
school
you
and
need
undergrad
to.
level.
That's why in most of the school, I don't know. I heard that in most of the school, they have the science club, own science club, and own discussion forum. they discuss about the possibilities of cross-linking the different portions of the science, and they can develop new techniques. But yes, that is required now, because everything is now in interdisciplinary. So you have to agree with that.
Okay, so if anyone don't have any questions, so like if anyone wants to conclude, Shubham, I think Shivaji sir is here. If he wants to conclude, or anyone can conclude.
Yes, sir is here. Actually, today our teachers are quite busy
Okay,
with regards to their work schedules and everything.
okay.
So we have our chemistry department, sir, head sir, my head of chemistry department, our very own, very favorite Shivaji sir. So, sir, if possible, then can we hear a word or two
So
from
like
you?
yeah. So yeah, first also give a kind of ovation to for
Yeah,
a nice talk
sure.
and a very excellent conversation.
Thank you.